Antiviral Therapies at the Center for Immunology and Infection Control at the Queensland University of Technology. So Dr. Idris is an antiviral therapeutics expert who focuses on developing novel therapeutic strategies to treat a range of viral diseases using gene editing and silencing technologies. He is an Australia Awards Endeavour Research Fellow, and in 2021 developed the world's first direct-acting antiviral siRNA therapy for COVID-19. So he has a PhD in cell biology and biochemistry from the University of Queensland. He has more than 100 publications, currently has a lot of funding running into the millions of Australian dollars. He is a fellow of the Royal Society of Biology at the UK, fellow of the Higher Education Academy at the UK, and a chartered biologist in the UK. So he's director currently of research at Intelligene, and this is a biotech company that develops RNA therapeutics against infectious diseases. So over to him. He'll be talking on developing RNA-based gene targeting against viruses, a new class of antivirals. Over to you, Dr. Idris.
Thank you very much for the invitation here. I actually enjoyed coming here. It's very quiet and serene, quite different to the VIT campus. VIT campus is great, but it's really quiet here, and I'm actually enjoying it. Feels like I'm back in the hotel room or so. But again, thank you for the invitation. And I was just reading the annual report downstairs, and it feels like I'm back home in the institute where I belong, right? The stuff that you guys do here is very similar to what I do. Sometimes when I go to conferences, I speak a different language. It feels like to a broad range of audience. I hope this language resonates more clearly to this audience than other places. Oopsie, here we go. There you go. Some disclosures. So all the work that I'm presenting here are not company work. And any opinions that I raise here are not the opinions of the company. Issues are global phenomena. It happens everywhere. Yes. He's so good at what he does. So this is my funding acknowledgements. So I'm funded by the Australian government, and also I work a lot with the Qatar government too for various things. This is an acknowledgement. So I like to acknowledge the people that do this work, the people in my group. Because without them, I'm nowhere. So I support the young early career researchers in all their endeavors, and they're the ones who do the work. I just tell them what to do and come up with the ideas, and they're really the ones who advance the science. So I really want to acknowledge all the work that they do up to this point. Here's QUT. So QUT is located in Brisbane. So Brisbane, Australia is in the east coast, right? So you've got Brisbane, Sydney, and Melbourne. So we're subtropical. So our summers are quite harsh. Probably arguably as harsh as in the Chennai area. So it does get really hot. The campus is located, we have multiple campuses. This one's located on the mouth of the Brisbane River. So you can see Brisbane River right there. It's connected by a CityCat ferry system, so you can go to different campuses and universities along the river. It's quite nice. We're hosting the Olympics for 2032, so please come to the Olympics and support India, right? Yeah, so or whoever you want to support. It's great. All right, so what do I work on as a virologist? I work on various things. I'm quite agnostic as to what viruses I work on. I usually have, I guess, a target of, which sort of viruses have an unmet need, a global unmet need. These are some of the viruses that I work on and have worked on. Majority of the time, we focus on respiratory viruses. That's where I started. I also have a key interest in zoonotic viruses here listed here. With my folks in China. I also have a history with oncogenic viruses, so human papillomavirus, Epstein-Barr virus. These are some of my interesting targets. And also tick-borne viruses quite recently. Our modality is the use of RNA to directly target the mRNA of these viruses. Viruses are great to target with mRNA, with RNA interference, because there's no off target, right? They're not like the host. You design it against a viral sequence. As long as it's not the same as retro elements in the host, then you pretty much only target the virus.
and they're amenable to these things. Obviously, viruses have anti-RNA interference mechanisms, but essentially we use not just siRNA, but we also explore other modalities like long antisense RNAs, which I'll talk about, which is not RNA interference, by the way. And we also tinker a bit with mRNA for various applications. And I guess the sole aim of this is to directly target these viruses. So we actually developed this COVID thing. So during COVID, we're like, you know what, let's use siRNA for COVID. That'll be fun. We applied for some funding from the government. Government, at that time, they were very generous. Whatever idea you have, well, this is an emergency. We're like, all right, cool. We'll apply. Got some funding, barely $1 million. We're like, let's do this cool project. Never been done before. And you know what? It worked. It worked so well. We're like, okay, great. What's next? And then suddenly the next day after we published it, and all these media outlets started getting crazy. Like, oh, world's first. So we didn't even know what was happening. And we're like, all right, so there's all this pressure now. The vice chancellor of the university is like, you guys should spin out. I was like, all right, cool. So we tried to spin it out in the US, and it didn't happen very well because we had venture capitalists already wanting to fund us for Series A funding for phase 1 GLP studies. And then guess what? Pfizer released the drug that same night. What's the drug called again? I forgot. It was a COVID drug. Anyway, it'll come to me. And venture capitalists are like, we're not interested anymore. Lesson number 1, you want to do something, do it quick, right? Secure your funding quickly, get the venture capitalists and delete all their social media accounts, and just secure that funding. So what we did was we weren't successful, so we actually sold the license of the drug. This is what Australian universities love to do, right? Oh, you can't come up with a spin-out, or we'll just sell your technology. They sold it for a good amount to a company called Gene Company. They're involved in a lot of RNA therapeutics, right? I didn't make a single cent from this, none of us. None of the co-inventors made anything from this, right? And then Gene Company then sold the license to another company called Intelligent, and a Taiwan-based company. They went public, and they became very rich after that, because there is an appetite for RNA therapeutics in the Asian region, and specifically in the North Asian region. And I know in India, there is that appetite too, right? Basically, the whole Asia Pacific, there is that appetite. So it's a good space to be in, right? It's the right ecosystem. So I currently work for Intelligent as a CSO. We traditionally use lipid nanoparticles as a delivery modality. I think one of the aims of the projects that we're trying to drive through now is, can we multiplex these various siRNAs targeting different viruses into one formulation? I mean, hypothetically speaking, it's a very easy thing to do, right? You just 1 + 1 + 1 + 1 = 4, right? That's the stoichiometry that you deal with. But it's never been really done before for multiple viruses, so we did that. But I don't want to talk too much about lipid nanoparticles, just to show you some example. We've developed so many different systems, and I'm not here to talk about these systems, right? There are different types, different lipid compositions, but bottom line is they all work. They all work quite well. We do a lot of animal work, and they're quite safe. This is one example where we actually multiplex. Don't look at all the data, but just look at here. So we multiplexed 3 siRNAs into a single lipid nanoparticle. This was a stealth lipid nanoparticle system, which we make by freeze-drying, tert-butanol type purification, not like the Moderna ones, very simple. And you know what? It knocked down viral infection in the mice quite well. So we're quite happy with that. And this is all intranasal. So we're interested in intranasal because we believe that we want to target the organs directly rather than inject it in the blood. Now, if you inject lipid nanoparticles, it will get to the lungs, right? Our original work with COVID showed that 27% of the biodistribution of these lipid nanoparticles actually end up in the lung. And that was enough to ameliorate, to decrease viral replication in the lungs. So that's 27%, right? But we said, how about we do it straight up the nose? There's another lipid nanoparticle system that we're working on, and this is a microfluidics device developed by a company called Pro Renata. So Pro Renata is an interesting story. It was my PhD student who didn't want to do a postdoc, opened his own company, got some angel investors. He said, you know what, I'm going to make LNPs. Because his whole PhD was all about making LNPs. He was loading like CRISPR stuff back then. So I'm going to make LNPs. I'm going to come up with this cool device because I'm sick of using nano assemblers, all these fancy machines with, hooked up and spend $250,000. So I'm going to make my own device. So he made this device. It's called Geno. So this device is quite cool. It's already patented, right? Essentially, it's like a cocktail mixer. You add this lipid to well A, add this to well B, and then you add this at this volume, give it a mix, comes out, you put it on an NTA or some sort of zetasizer, measure your particles, and guess the peaks are very tight. I was like, no, that can't be right. Can't be that perfect. But he's finessed that in such a way that that his company has become quite successful. So it's an Australian company. He opened a subsidiary, moved to Qatar, and he just secured all these fundings from the Qatar government. And I'm like, I should go to Qatar and actually start my next company there. So he's doing really well. Next year he's going on to GMP phase. So basically what I'm saying is we compared his LNPs to stealth LNPs, so SLNPs in red. And here is a biodistribution study showing the bioactivity of the siRNA. In this case, we use beta-actin in mice. You can see it performs quite well, quite comparable to, I guess, conventional lipid nanoparticles made by nanoassemblers, right? You can see the knockdown at 24 hours, it peaks, whether you nebulize it or you give it intranasally, it doesn't matter. Now, one important point, you nebulize lipid nanoparticles, a lot of lipid nanoparticles, their distribution goes from that to like that, because there's a lot of aggregates that break down, but his one just stays sharp. I'm like, what is up with this device? There's something special about it. And obviously it's proprietary, you can't share anything with me, but all I'm saying is we're tinkering with that technology. Now this is an incomplete slide. We're also tinkering with a hydrogel called Sol-Gel. It's a thermostatic hydrogel in collaboration with the University of Queensland. The idea behind this was, I went to the chemist, to the pharmacist, I said, look, I need something that stays in the nose. Those who are virologists know that respiratory viruses start in the upper respiratory tract. And once it goes to the lungs, then you get hospitalization, those sort of problems. Right? So I thought, how about we target it while at the early stage? So I said, but I don't want to put LNPs up there because it doesn't last long. We've done biodistribution studies with intranasal, last 24 hours max, and then it just gets cleared, right? Because it's quite external. So I said, the Sol-Gel will be great. So the Sol-Gel is a thermostatic Sol-Gel. It gelates at body temperature, meaning that it goes from liquid, and then once it hits body temperature, it becomes like a gel. So you can see there that Sol-Gel, in this case, is green in color. We can actually make all sorts of colors, all rainbow colors if you like. So you can see it stays there. Now, I have some biodistribution data which I haven't put. I'm still waiting for the data from my postdoc this afternoon. But trust me in saying that the siRNAs linger around up to 72 hours. These are unmodified siRNAs, right? The controlled release into the nasal cavity, nasal epithelium is quite impressive with these soldiers. So that's one of the projects that we're tinkering on, possibly as a way to keep something there. Then you use less doses, right? We also tinker with chemically modified siRNAs. Here's another example. So we want to move away from using lipid nanoparticles. What do? You just chemically modify it, right? Here's the chemical modification with a cholesterol at the end. This is an Alnylam trick. Cholesterol modified siRNAs can go to the lungs very well. You do an IV, as you can see here, right? So that's the lungs. And the signal compared to unmodified ones, the signal just stays in the lungs, right? It's great. So if you don't want to use lipid nanoparticles, you have to chemically modify that. But there's a cost involved. Those of you who do this work already know there's a cost involved, a dear cost involved to modify these siRNAs. You can also intranasally deliver them, but it doesn't last very long. If you deliver naked siRNA into the lungs, after 24 hours, you won't see a signal. So if you're developing a drug, an intranasal drug that is unmodified, not encapsulated lipid nanoparticle, you're going to have to do a daily dosing, right? Inhaled or intranasal. But if you chemically modify it, it will go past up to 72 hours, which is what you want. Okay. Another siRNA technology we're tinkering with is called NUCM. So siRNAs, as you know, act very well in the cytoplasm, but it can also act in the nucleus, right? This is very old literature. AGO2 which is part of the RISC complex that allows for the chewing up of RNA present in the cytoplasm. But AGO1 is in the nucleus. A lot of people don't know that or underappreciate that. Very old knowledge, right? So we thought, how about we make an siRNA that can go to the nucleus also? So dual compartment targeting, right? So all we did, and this is IP protected, I can't talk too much about it, but essentially we've done this for a few years now. It's an entry motif that we put as an overhang at the end of the siRNA. And I can't tell you which position either, but essentially it looks somewhat like that, and it allows the active transport of the siRNA into the brain. Now, most siRNA experiments, what do? 30 nanomolars, right? I'll get you to cytoplasm. If your RNA target, whether it's a link RNA or whatever, it's in the nucleus, how much do you add? 100 nanomolar? Now, translate that into a mice. It's almost subtoxic. It's a lot, right? 3 times more. In the mice, you do what? 0.5 milligram per kilo, right? IV. You triple that, your cost triples, and you do your GLP toxicity study, and most likely you'll see some issues, right? And there's a cost involved. So we thought, we improved this. Now, influenza is a very interesting model because it's one of the only viruses, only RNA viruses, that actually goes to the nucleus, i.e. the genome gets moved to the nucleus. It replicates there and gets shuttled out into the cytoplasm. It's a very unique virus. So we thought, let's use influenza. Because influenza siRNA targeting is very tough. We can never get above 80% knockdown, never. And we're like, why? We've designed hundreds of siRNAs, quite literally, and it's always 50%. So then we did it. And here, when you apply Nucum to one of these poor performing siRNA, you can see enhanced effect. And no, it does not. So these Nucum siRNAs do not induce immunostimulation. So we've already screened for that, but it doesn't evoke interferons or NF-kappa B. So that's already done. You put it in vivo, you can see that improvement there in influenza. You can apply this beyond viruses too, for example, to certain nuclear targets, right? So in this case, we use U6 small nuclear RNAs. You can see Nucum targets it quite well compared to a conventional siRNA. These are nucleus-specific ones. They don't shuttle back and forth cytoplasm, they're only the nucleus. Here's another one we're tinkering with as a master's project, where we target a link RNA that controls LDL receptor. LDL is important for sequestering cholesterol. Got nothing to do with virology. We thought we'd just have fun, right? And you can see Nucum somewhat improves that, right? This is all very early data. Here's one here. HTLV1 is a virus just like HIV. It's a retrovirus. It will integrate into the host genome and cause a cancer. It's a T-cell lymphoma. It's very prevalent in indigenous communities in Australia, Aboriginal indigenous communities, and also in Japan. In Japan, it's almost endemic, right? Millions of people have HTLV1, but only 1% or 1 to 2% are symptomatic, right? But it's a serious disease, unmet. There's no drugs for it. But the important thing is, siRNAs are known to trigger what's called transcriptional gene silencing. So this is very old literature, right? siRNAs will go into the nucleus, they engage with a target that could be a link RNA inside the nucleus, and this siRNA actually stop the chromosomal rearrangement, and it silences the genes, right? And it just stays there through a molecular cascade, which I won't describe now. Now, Nucum can improve that. So in the last figure, if you treat, this is the knockdown of the HTLV1 link RNA, and if you add epigenetic inhibitors, so in this case 5-AZA, right, cocktail, you lose that, which means that it can induce some sort of transcriptional gene silencing, but we're trying to confirm this with some chip IP assays to see whether this is happening at molecular level. So we claim that our siRNAs could fill this void, right? We've got antisense oligos in the market, but it does not induce transcriptional gene silencing. As you know, antisense RNAs, they work in a different mechanism, right? They use RNases and all that, RNase H-mediated degradation and all those sort of things. And the problem with antisense oligonucleotide, they shuttle between cytoplasm and nucleus. People don't talk about that, but they do shuttle. I've got a guy at Imperial College in London who's telling me every time I put anti-sense, and he's a chemist, because every time I put anti-sense, I see you go in and out of cytoplasm, but no one talks about it. So there's constant flux, right? So that's the issue. Right now, I'm planning some monkey primate studies for non-GLP cell. I'm trying to spin off this technology soon. So we already have a GMP partner. This is the device that I'm thinking. I actually met these guys at a conference in India a few years ago. Aptar, they're a global company, but they have a branch here. And I sort of like the device. I'm like, maybe I can use that as an inhalable, right? So that's what's happening now. I want to talk about EVs, move away from lipid nanoparticles. So EVs is something that I've been quite passionate with over the past few years. EVs are amenable to engineering. So a lot of people either engineer or just add things like quick chemistry or just pretty much mixing 1 + 1 equals 2 type of thing. So we've done this for quite a while. You can put all sorts of cargos, protein, nucleic acids. You can put functional moieties on the top by various ways. And I just want to show you a few examples of what we've done. Here's one example where we put H2 protein, quite literally expressed protein, on the surface of these EVs. So this is by molecular engineering. It's work done with Koch University, and you can see it neutralizes, because A2 binds to SARS-CoV-2, right? It's its receptor, right? So we thought, oh, maybe we can neutralize the virus, and indeed it did, right? So neutralize the virus. Positive control is virus-like particles. These are VLPs. Obviously, they're going to work, right? So that's just one example. Here's where we actually engineered cells. So in this case, HEK293 cells with EVs containing nanobodies. So nanobodies are like a small version of antibodies, if you like, right? This is the VHH, right on the surface of the EVs. So we argue that if we have multiple nanobodies on the surface of these EVs, we can have a nice, very potent antiviral effect. And this is just in vitro showing, that we can actually, compared to the control, can actually neutralize various different variants of SARS-CoV-2. Now, we put this into animals, and we decided to move into neural stem cells. And why? Because neural stem cells are purported, and we actually show that it's anti-inflammatory. Now, a lot of things in the literature say, oh, these EVs from these cells are anti-inflammatory. I suggest you check. Suggest you do those experiments yourself, because we've done a lot of it, and a lot of them are fake. Here's lesson number 2. Do not believe everything you see published, right? So this is for the young people. Always verify yourself. Just do not believe it. So neural stem cells, we did the same engineering. Intranasally, when you give it, it goes to the lungs quite naturally, but here's the problem with EVs, right? 24 hours, it's gone. It gets taken up, gobbled up, it's natural, doesn't stay long. So you got to keep giving it, right? If you're doing dosing, so that's another lesson. The view of these EVs, it doesn't make them sick, so the mice seem to tolerate these EVs, right, intranasally. We did a panel of just an anti-inflammatory nanostring panel in SARS-CoV-2 infected lungs treated with these EVs, and you can see a transcriptomic change or deregulation of certain inflammatory genes, which actually confirmed that these EVs are actually somewhat anti-inflammatory. So that was quite nice, right? So when we did this dosing, we thought, at this stage we ran out of funding. We're like, I can only afford to do 2 doses. But we decided to do sparing doses, right? It did have an effect, but we argued that if we did it daily, we would see a better effect. If you remember, by distribution, 24 hours is gone. So we're seeing a very minimal effect here. Those who have a knack with NTA, that's the image there, and so forth. So that's just another example. Here's another example where we put a full antibody, FCFV antibody, targeting a virus called EMCV. This is a pig-borne virus, very prevalent in China. It's no longer a problem in a lot of places, but in China it is. Gave the EVs into mice infected with EMCV. In a mouse model of EMCV, it's 7-day lethality. So 7 days, these animals are dead, right? Body weight loss by day 6. By day 7, they're conked out. These mice completely rescued, multi-organ attenuation of full multi-organ viremia, which we've never seen before, right? Heart, brain, you name it. So that was a nice study that we did. Here's another example where we tried to load it by molecular engineering with antisense RNA. Now, antisense RNA is a very old literature. Essentially, it's around 100 to 200 base pair. The idea is that it will bind by Watson-Crick base pairing to the mRNA, and it will degrade that mRNA through mechanisms unknown to anyone. But it's been done before. Why do we do this? Why not use siRNA? Well, if you're dealing with a virus that mutates like crazy, like all RNA virus, you want to use a potential drug that can target a big region, and if there's point mutations anywhere else, the Watson-Crick base pairing is still going to be strong, right? If you use siRNA, 23 base pairs, it can probably tolerate 2 mutations, right? Depending on which position, then the drug loses efficacy, right? Unless you multiplex it. So here we use molecular engineering using a system here where we tether the antisense to an RNA binding CD box. That CD box will bind to L7A, which is an archaeobacterial protein, and it will load it tethered to a CD63 transmembrane protein. So essentially, you've got an EV with a little wiggle inside. So it will load only inside of the EVs. And when we give it to mice, it somewhat works. And this is IV, right? This is, intranasal. So this was intranasal. It was IV. Straight by retroorbital injection. It's a daily dosing, right? You can tell we had a bit of funding here. We could afford daily dosing. So it sort of worked, right? So that's just one example. We also like to try loading mRNA by molecular engineering. So we thought, let's pick a difficult one. So we thought, how about we load an mRNA, but that's only restrictively translated in a particular cell type. So at this time, I've always been interested in T cells, number one, because they're hard to transduce and transfect, right? Everyone knows who works with T cells, they're almost impossible to transfect, right? So we looked at this old publication and we saw, hey, there is a certain region in HIV, the 5' untranslated region, right? If you truncate it, because HIV infects only T cells, right? And if you truncate it, which looks like that, the transgene expression of the truncate of the IRS activity, which is the IRS, which is at the end of the HIV, is enhanced. We're like, using this knowledge, how about we put that truncated 5' UTR, shove it right in front of the GFP mRNA coding sequence. And that's what we did. So I told my PhD student, okay, make some EVs, engineer it, using the same thing, CD63, right, L7A, same thing, right? Put it in EVs. And then I want you to plate out T cells, HEK cells, and liver cells, have G2. Let's see if there's restrictive expression of mRNA. And guess what? Only in T cells. So the truncated version seems to work. We're like, great. Okay, what's next? So next I told him, how about you put some T cell targeting just to make it a bit more specific, right? So he made some EVs that have a protein expressed that binds directly to CCR4. CCR4, which is highly expressed in T cells, importantly in HIV-infected T cells, right? So I did that, and I told him to do a flow cytometry after you do your binding assay. And guess what? Only in T cells, right? Highly restricted in T cell expression, not in THP1, not HG2, and so forth. So that was quite nice. So I told him, how about you play with one molecular engineering approach? So there is an aptamer called C16, which is specific to T cells, so CCR4, CCR5. So we thought, okay, let's synthesize this, get it ordered, shove a lipid at the end, and put a FITC there. Now, this is an old trick, right? It's an old trick. If you want to put aptamers and load them onto extracellular vesicles, you just put a lipid at the end. So I told him, put a lipid, and it should look like that. But in actual fact, it should be inside and outside. You cannot control the loading of proteins in this method by having it all outside. Some of them are just going to go inside the inner leaflet. We did that. I told them, put it on T cells, and you can see we tried 2 lipids, DNPE, DSPE, right? And you can see that T cells are positive with FITC are in T cells. So this is ongoing work. What else are we doing? So we're also loading short hairpin RNA. Now, you can't load siRNAs by molecular engineering into EVs. You just cannot, right? You have to do it in the form of a short hairpin RNA. In this case, what we've done is that we co-express AGO2, which binds to short hairpin RNA, right, inside the cells. No transmembrane protein, there's no CD63. And we then purify the EV, put it onto cells infected with HTLV1. So these are very hard to find, these cells, right? And I told her do a knockdown assay to see if your EVs knock down the shRNA target, in this case, text mRNA, which is one of the genes that the link RNA of the HTLV1 target is involved. You can see it knocked it down. So this was quite nice. Next, I told her, how about antisense RNA? So we did the same thing, antisense, targeting the long coding RNA. And again, she's getting a very good one, good knockdown there too. So that was quite nice. We also quite interested in targeting the brain. We were just having a discussion with the professor here earlier about brain targeting. So how about we know that EVs can go to the brain. You'll see so many publications, this one goes to the brain, right? But it's not effective. One thing that these papers don't tell you is how efficient it goes to the brain. A lot of the time, it's a very passive process. You put EVs intranasally, and then you do 24 hours staining, you'll probably see EVs in the brain. But is it enough therapeutically? The answer is most likely no. It does go into the brain. So in this case, we thought, how about we modify these EVs with a transferrin receptor and or ApoE? Knowing its biology, it should flip into the other side of the blood-brain barrier. It gets taken up and goes to the other side. This is our hypothesis, right? So we spent months making stable cell line. We're like, if we're going to do this, we're going to do this properly. We're going to do stable cell line, do single cell selection, drug selection, right? I want pure clones because when we get these stable lines, then you can load whatever RNA you want, mRNA, antisense, SH, whatever, right? But I don't want to keep doing this. So we developed stable cell line. After 6 months, you can check after I don't know how many passages, the transgene is now stuck inside the cell. So we're like, great. Now we can actually ship them to other collaborators that can use whatever they want, right? Did this, check the NTA for EVs, they all look good. Nice particle numbers, these cells are not sick. So here it is. So we thought, let's try the easy one. Let's go send it intranasally into the brain. You don't really need blood-brain barrier molecularly engineered EVs to go to the brain if you send intranasally. If you've got this olfactory nerve that will just take anything, well, almost some things into the brains, and it bypasses the blood-brain barrier. So we thought we did the easy one. That's the lungs, intranasally, obviously, it goes to the lungs. But the brain, we're not seeing the signal. We're like, oh, so there could be aggregation, right? These EVs that have all these things on the top could be aggregation, can't move up. But we do see a little signal there. We're like, okay, maybe it's gone up there. So I told to file on postdoc, I said, how about you shove it into the lungs? By IV. So we only used the transferrin receptor positive ones, and after 24 hours, you start seeing some of the signal. Right? We're like, oh, great, good, but still not good enough. So we're still trying to improve this and trying to understand. So we haven't started loading these EVs with RNAs yet because we want to finesse. The system and make sure what we're seeing is real and not just some passive effect. So right now we're setting up transfer co-culture, blood-brain barrier in vitro models just to understand the mechanics of it. Did you know that if you nebulize EVs, they still look fine after nebulization? Unlike lipid nanoparticles, which sometimes get damaged, these EVs look good. So that's pre-nebulization, right? So that's the NTA profile. And after, it looks all right. Like for a guy that looks at nanocyte data a lot, I'm like, that looks good. And then if you do the nasal cavity in the lung, and that's 24 hours, it's still there, right? So we're like, okay, maybe we can start tinkering with that. We also interested in other modalities. Here's a carbon nanodots. Those who are material scientists. These carbon nanodots have been functionalized with aminophenolboronic acid, which binds onto the glycoprotein face of various viruses. And here it's broad acting against a whole lot of viruses. I just wanted to show you, we're also tinkering with other things. Here's my final slide. I thought I gave you a flavor of what I do in the lab. I don't want to talk deep into the data we have, but essentially we're interested in loading all sorts of things into EVs. We're also interested in trim-away mRNA technology. Those who do not know, trim-away is a bioprotect system where it's just an mRNA that encodes two things: a nanobody or you can put a peptide if you want, and a TRIM21 protein. Now, why TRIM21? Because TRIM21 gets taken up by a proteasome in those cells. Now, if you put a nanobody on top that targets an antigen of interest intracellularly, think about it for Alzheimer's disease, Parkinson's, right? For like amyloid plaques or something. You could use this technology to remove insoluble proteins and think about diseases like Kuru, prion disease, for example, right? This would be great for it. So we're tinkering away with a bit of trim away mRNA technology. It's a hard project, but I think we'll get somewhere with that. And we're also exploring carbon nanodots as a potential platform. The reason being is that I told my friend here, I said, look, can you make something that looks like that? And he's like, yeah, I can sort of make that. These chemists are like, yeah, we can do that. We'll do it, whatever you want. And so I said, let's check the stability. So if you put siRNAs in, expose it to serum at 37 degrees Celsius over time, it will degrade, right? You all agree with me. But when you put carbon dot complex siRNA in there, for some reason, it doesn't degrade. We believe that these carbon dots, because of the charge, somewhat is stabilizing the OH backbone on these siRNAs. We're like, this is great. All I have to do is just get some carbon dots, which, by the way, he made it in bamboo as a carbon source. Bamboo, you can do citrus. It's great. It's an underappreciated field. Like, it's very green. It's green science, right? Just add synthetic RNAs there. It'll just mix with it. There's no fancy chemistry, right? Because the charges are different. And it works. So we've got another research project on that. Very last slide. Acknowledgements to the people that are involved in the work. I've got great folks working on these projects. And some of my students, actually a lot of them are from India too. So when I come here, I'm always looking for talent. I just come and say, hey, come to my lab. The last girl I brought in, she came from Bangalore. I'm like, hey, you're coming with me. It will give you a scholarship. You're that good. So I'm always looking for talent. Thank you for your time and listening to my waffle. Thank you.
Yes. PDI is different between the nanoassembler to our device. What is the difference between when you prepare nanoparticles, lipid nanoparticles?
So it's a microfluidics device, just like the nanoassembler, right? But it doesn't use power. It's just check cocktail thing. So there's a series of tunnels, which I still to this day do not understand how it works. But when you take it out and you measure it, the charge, it looks nice. For some reason, it just magically makes that. And right now, they're going on a GMP stage for next year. They're going to be the next CMO, contract manufacturing organization for that region.
You mentioned that distribution is narrowed.
Yeah.
So there, the PDI must be less. Like what significant it is when compared to nanos?
Yeah, so I can show you some data after this. I need to dig it out, but if the nano-assembled one, it's nice, but the genome is like almost perfect. I'm like, that's really unnatural. So, it's really good. Obviously, even though he was my student, he wouldn't tell me how this whole thing works. But but it's great. We're happy to make some for you guys if you want to try it, and we'll just ship it over, right? Dry out shipment though, unfortunately. Unfortunately, I can't pass the device around. You have to sign some sort of MTA or something. But yeah, more than happy if you want to play around with it. Yeah.
Hi, sir. So I have a few questions.
Yeah.
So one is regarding the epigenetic silencing using the siRNA.
Yeah.
So how long it stays after binding?
So there is a molecular cascade that happens, which I didn't want to talk about. So there's a bunch of proteins, ones that engages with AGO1, potentially also AGO2. There's also AGO2 in the nucleus, by the way. And there's other proteins that actually bind. If your target is a link RNA, right, in the chromosome, it's link RNA is what do they act as? molecular sponges. They could act to, shut off promoters, all sorts of things, right? Target that. The conformation of the chromosome stays there, right? That's what siRNAs do. And people have forgotten this. My friend Kevin Morris, right? He's the one that discovered this phenomenon back in 2001, I think. He discovered it. And he told me a story. Is that I went to a conference in the US. I was like happy about this technology, right? I gave a talk about transcriptional gene silencing, and then Jennifer Doudna walks into the room and talks about CRISPR. And then my career was over, he said. It was supposed to be the next big thing, right? Transcriptional gene silencing, but then CRISPR came about, and I just lost attention. But apparently Jennifer Doudna is a very nice person, he said. Yeah. Yeah. But yeah, so there's a molecular cascade that occurs that's already published. You can follow it, but it's quite complex. What it does is it locks it in conformation.
But that stays for how long?
So it's purported to be semi-permanent. How long? Don't know. But it's long. Long as in probably, I don't know, in vitro, 48 hours, based on Kevin's publication, 48 to 72 hours. Yeah. It stays quite long. I haven't tested it in animals yet. That's the next thing I want to do. Yeah.
It's basically blocking the chromatin at that region.
Absolutely. Absolutely. It's pretty cool. And people have forgotten about it, Yeah.
Hi. So what really caught my attention was the EVs that you're using for your crossing of the BBB.
Yeah.
So what I'm thinking of, or what I'm really curious to know is that first, once you're delivering your drug, for example, or siRNAs, so there is a lot of, your experiments you've done, there are a lot of experiments done in the HEC tonight. It seems to be a little funny cell line because
yes, it is.
When I'm a very sort of like a cytokine, chemokine guy.
Yeah.
So when you're specifically measuring the mRNAs or whatever, screening for the inflammatory response,
Sure.
the EVs,
yes,
you can get a lot of varied results with HEC tonight.
I agree.
Now, do you actually have you done some work in the primary cells as well? Because that's the crux.
Yeah, so the anti-inflammatory ones were done on primary neural stem cells.
Right.
The NSCs, I think they're A4 cells, were donated to us. You know what? We've never done anti-inflammatory testing in vivo for HK293s. We've never done it.
Or the inflammatory as well.
Yeah.
Because there is a reason why BBB doesn't allow or has to keep the environment sterile as long as possible. So this is one. Second is the longevity of that particular siRNA in the EVs across the BBB.
Yeah.
Because there is a very important reason why physiological conditions don't allow things to.
Absolutely.
So that's a very, 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 as far as when it comes on to patient studies.
Absolutely. Look, EVs can go to the brain. There's no doubt. You can get any cell line. There's school of thought that EVs from a particular organ has a tropism to go back to where it goes, and that's why we started with neural stem cells, right? Yeah, it goes in there, but not much. We see a diffuse pattern. But you're absolutely right. I think our biggest concern is not it going in, keeping the RNA for long amounts.
Yeah.
The stability is very hard, and the company I work for as a CSO, this is a technology that we're developing, which I can't talk about, but I'll tell you now, it's hard.
You can infect H293, keep them as long as possible when it comes to primary cells.
Yeah, it is.
It's a different ballgame.
Yeah, and it's costly with primary cells. Yeah, you gotta add insulin, all sorts of recombinant this, recombinant that. Yeah, it's a finality is hard. I mean, proof of concept, we can do that stuff. But as a final product, that's my mission for the next 5 years. Yeah.
Have you also been trying synthetic peptides?
Synthetic RNA loading?
Yes.
Yeah, we've tried that before. Electroporation, lipo, you name it. Look, the experts in that field are the Korean groups. So if you go to like EV conference, you always see this Korean cohort. They tend to be from the same university, and they're very good at it. We electroporate this, and this is how many copy numbers of RNA are in the EVs. They're very good. But I said, is it scalable? They said, oh, not really. Batch-to-batch variation, but you can. And you'll get probably 1 copy of RNA per EVs. Molecular engineering, I'll tell you now, you'll be lucky to get 10 to the -5. A lot of EVs. If you have 10 to the 10 EVs, 1 dose, you only have 10 to the 5 EVs that are positive for your RNA cargo with the API, your active pharmaceutical ingredient and stuff. You need to put a lot in there. Yeah, big limitations. That's why I'm doing it. I don't work on easy problems. Yeah. Yes. Is that? Oh. Sorry? You mean viral
envelope
on the EVs? No. Or you can engineer it. So I know people in Utrecht University in Holland, they put VSVG. It's a good trick, but you can't go to clinic with that because it's a viral protein. But it's a great trick. Yeah, you can do that. I've seen that paper. Are you asking whether I've done it before? Are you asking whether I have used? With viral proteins, I've never done before, but it's very easy to do, and it will work. Yeah, surface engineering, there is less of an evolutionary pressure for the cells to make it. When you load things like RNA, there seems to be a pressure because cells don't like having foreign RNA, right? Your RIG-I, your whatever, O2AS synthase receptors, they all want to counteract having foreign RNA in there. Yeah.
Hello.
Yes.
So I'll just extend the question. So how long your siRNA is stable in the EVs?
After you purify it.
After you purify.
We're doing those studies now. So we're doing up to 3 months, different formulation, lyophilized, storage in -80. So we're still doing that. Right now, after 2 weeks, it's still there. All right, it's still there.
2
but
weeks when you store it in 4 degree or
powder. Yeah, still there. Not good enough for us. Yeah, we want it longer. I think this is the limitation, storage-wise. To avoid batch-to-batch variation, I suggest keep it in liquid and just store it. It'll probably stay longer in there. That's my opinion. But I can't give you that data now. I just don't have it.
You just now said that out of 10 to the power 10, 10 to the power 5 is probably the actively loaded EVs, but that's a big estimate. You calculated it in that way, or?
Oh, it's not an estimate because I've done it a lot. So 10 to -5 is probably the best you can get, depending on the RNA. Like mRNA does not look like a viral RNA. You put a long antisense RNA, it looks like a viral RNA. So my hypothesis is that certain RNAs that mimic microbial RNA will probably not be kept long enough in cells, whereas mRNA has got a cap, either poly A tail, looks a bit more natural, So maybe the cells have a preference for that. I don't know.
But the purity of your cargo-loaded EVs also depends on how you purify it, right?
Of the cargo? Oh no, the purity is the EV purity when you run through the active flux or whatever, your TFF. It's not going to be pure RNA inside the EVs. There's going to be microRNA. It's going to be a whole lot of stuff there. The intention is not to clean up what's inside. Actually, if you want to translate these EVs, it's very important that you remove things on the outside of the EVs. One of the biggest problems with EV purification, doesn't matter what you do, is the DNA. FDA dictates you need to have around 10 nanograms, less than 10 nanograms per microliter of DNA, something. I'll tell you now with these EVs, purified, looking at 2,000 nanograms. So you got to put a lot of denarase, DNases, To me, that's a bigger problem. The RNA will stay in there. Yeah.
Just want you to discuss more on the shuttling phenomena. So even when you're loading in nanoparticles and putting it inside the cell, then also that shuttling happens between the nucleus and the cell.
It does, yeah.
Even if it doesn't have something that takes it to the nucleus.
Oh, you mean with lipid nanoparticles?
So is it active shuttling?
If it's active shuttling, yeah. So we're doing RNA co-IP
molecular studies now to determine. I can't tell you what the protein is. That's something we're going to patent. But there is a molecular pathway that involves the active bringing in. So we picked the motif specifically because we think there is a cognate partner. So it brings it into the nucleus actively. I just don't have that data to show you now. But yes, it just does get taken in actively.
So when the tag is not there, have you observed any passive shuttling?
Of course, yeah. That's why I gave the example, 100 nanomoles, 30 nanomoles. They'll always get passive shuttling of siRNAs, but there is significantly more siRNA being actively brought in if you have the NUCM technology.
One last question.
Yeah.
So I saw when you were doing it intranasally, the dose was 10 to the power 8.
Yeah.
When you went IV, it was 10 to the power 9.
Yes.
So how did you calculate the dose? You scale it 10 times?
We didn't. We just guesstimate. I closed my eyes and I'm like, the liver's going to eat all my EVs. So I told my process development team, can you make more? That's my science, right? Because I know the liver. I don't have a stoichiometric calculation. Generally, I do at least 10-fold more. If not better, you do 100-fold more because you're going to lose everything in the liver, I guarantee you.
I think we'll stop now. And thank you, Dr. Idris.
Thank you.
I